All right. So at this segment, you are looking at artificial intelligence in machine learning, computer-aided drug design, artificial intelligence in machine learning through computer-aided drug design. You know, from the word computer-aided drug design. If you remember what we did in the last section, we are saying computer thinking in the same manner with men. Now, we are now bringing that computer now to come and assist us in designing a drug. So we may ask ourselves, what is a drug? I know for majority of us, especially people in biological science, and at least many of us will have something to say about drug. And I can say drug is a medicine or any other substance that has physiological effects when ingested or otherwise introduced into the body. A drug is any substance that has physiological effects when ingested, either orally, intraperitoneally, or through injection into our body. It has physiological effects. For example, somebody is complaining of headache, you take Tylenol and the headache goes, that is a physiological effect. Somebody is having stomach pain, he or she takes a medicine and that stomach pain stops, that's a physiological effect. Someone has COVID and it takes some anti-COVID drugs and that COVID is no longer there again, that's physiological effect. So any substance that we take in, either orally or through another means, that has physiological effects are referred to as drug. And we use AI to an aspect of AI called computer-aided drug design to design these drugs. To talk about what is drug design. Drug design is the inventive process of finding new medication based on the knowledge of a biological target. So drug design is not like we are designing an apartment, no, a building. It's a process of finding new medication based on the knowledge of a biological target. What that means is, if you want to develop a drug that will cure COVID, you are expected to have a better understanding of COVID. How does COVID work? How does COVID get into the human body? How does it perform its destructive function? Once you understand that, it will help you to put together a process to find new drugs or new medications, you understand, to target the COVID, which is the biological target, you understand? So that is the process of drug design. And another way, it involves the design of molecules. And in this case, when we're talking about molecules, anybody that is not a science student, just think about a drug. Let's put it that way. When we are in the process of developing it, we refer to it as molecule or ligand. When it has been approved by a regulatory body like FDA, that's when we call it a drug. So anytime you see me using molecule, ligand, I'm referring to drug. You understand? So this drug design involves the design of molecule compounds that are complementary in shape and charge to the biological targets with which they interact and therefore we bind to. What that is saying is, when you are designing a drug, for example, when you want to design a drug against SARS-CoV-2 spike protein, the SARS-CoV-2 spike protein is used by the spike virus to gain entrance into the human body. When you want to design drug for that target, I will be using COVID as example. When you are designing a drug for that, in drug design, it is advised you understand your biological target. In this case, the spike protein. You understand? So when you understand the spike protein, you know the spike protein, you know the different amino acids present on it. You look at the shape of it, then you'll be able to go and design a compound, a drug that will complement the shape of that drug, of that target. Are you guys listening to me? So once you do that, you are doing drug design. So Drug design, this molecule that you're talking about, the drug, will interact. If you look at this diagram, if you look at this structure you have here, this is a drug on the market. It could be paracetamol, it could be Tanolol, it could be metformin, it could be any antiretroviral drugs. This is the biological target. Let's say this is the spike protein. We have this, let's say that's the spike protein. You have your drug binding to it. That is the way it binds. You have the drug binding to it. Once it binds to it, it will interact. There must be interaction between your drug and the biological target. There must be interaction between your drug and spike protein. It is when the two of them bind and interact together. That is when the spike will not be able, the COVID will not be able to infect another person or gain access into another person's body. So there will be interaction between drug and what? And the target. So when it binds, the drug will either inhibit, like maybe, for example, you want to stop COVID from gaining entrance into a human body, you give a drug to COVID so that the drug will inhibit, it will stop COVID, the spike from recognizing the protein on human cell to infect, or some drug will activate. Maybe there are some diseases whereby some enzymes in their body are not functioning. They will say, okay, go and take a particular drug. What that drug will do, the drug will activate the enzyme that is not working so that it will be able to perform its biological function. You understand what I'm saying? So when we're talking about, so there are selected design molecules. There are a lot of molecules, organic small molecules, compound, complementarily shaped to the target, oppositely charged to the biological target. So when you design drugs, you take a small molecule, and what the mean by small molecule is all the drugs you are buying on the market. Most of them are small molecule because their molecular weight is small. That's why they refer to them as small molecule. But when you are talking about vaccine is not a small molecule because most of them are protein structure. So anytime you hear us talking about small molecules, we are talking about drugs with smaller molecular weight. That's what we mean. So most of these drugs, they have shape, like we are going to see by the time we start the practical part of this workshop. We do that, then they are charged. As you can see in my diagram here, all these things, they have charges. Don't forget your element has charges, and hydrogen is positively charged, sodium is positively charged, chlorine is negatively charged, because all these elements also make up all these small molecules. Carbon, this in red is oxygen. All these ones, this one in gray is carbon. You understand? We have many of them. I think this should be, maybe this should be fluorine. This one should be chlorine. We have different charges. So because they have elements, the drug have elements, they also carry charges. So when you are designing your drug, you have to consider the charge on it. And by the time we start designing our own drug, during this project, in this workshop, you will see us adding charges to our drug we are designing. So that's the way it is. So now, we now talk about drug design frequently, but not necessarily we like. Most of the time, most of the drug we do these days, it's not necessarily, but majority of them depend on computer modeling, computer algorithm, because there are models that has been developed that can think more than what you can think. All you just need to do is give the information to the computer algorithm, and to give you information that may take you years of experiment to achieve. That is what they do. So this type of modeling is sometimes referred to as computer-aided drug design. So when you make use of computer to design your drugs, when you make use of computer to do a lot of thinking, a lot of design, a lot of predictions in the purpose of making drugs, that is what we refer to as computer-aided drug design. And most of these computers, they make use of artificial intelligence softwares, artificial intelligence programs. And by the time we start this workshop properly, our practical aspect of it, we are going to do installation of softwares. Everybody, you will let me, if we are using iOS or you are using Windows, I will know we are going to install softwares. You understand? And some computer-based algorithm. So when you make use of these softwares in designing a drug, that is when you are using computer-aided drug design. Look at the name computer-aided, computer-assisted you in designing a drug. So what is computer-aided drug design, which is called CADD, C-A-D-D, represents a computational method or resources that are used to facilitate the design and discovery of therapeutic solutions. The computer, they are used to facilitate. Ordinarily, if it takes you 10 years to design a drug, if you use computer-aided drug design algorithm, it will take you either half or less than half of the year you spent without using computer-aided drug design. So, and drug design with the help of computer may be used at any of the following stages. There are different stages we use it, either to identify its identification using virtual screening. You can, okay, if you have thousands of drug compounds, you can use computer to say, okay, out of these thousands of compounds, which one is likely to cure COVID, for example. You get my concept? Another thing is it to lead identification. Okay, if you have a particular, okay, out of 10,000 compounds, you have 2 compounds that is likely to cure COVID, but you observe that that compound cure COVID, but also causes other damage to the patient. We can use computer aided to design, and we have computer aided, we use that CADD to remove whatever property on the drug that causes damage to the patient, and we will leave the property that cure COVID. We use computer-aided drug design for that. So when you have a lead drugs that cure COVID and still causes other damage to the patient, we can use computer to take away whatever functional group on the drug that causes damage to the patient and leave the functional group that cure COVID, because our purpose of designing a drug is to cure a disease without causing another damage. Don't forget. And that is, there are a lot of drugs that could not get to the market. Not because they are not working. They are working, but because they cause more damage, even than the disease they are sent to cure. Imagine when you have a drug that can cure COVID, at the same time can cause cancer. You, instead of you using it, you will rather step it down. So we use computer-aided drug design to do lead identification and lead optimization. Optimization for other pharmaceutical properties while maintaining affinity. We optimize the properties. If there are some drugs that they are not soluble in water, they are not soluble in lipid. And when a drug is not soluble in water and lipid, it will be difficult to exert its therapeutic effect in a patient. So we do a kind of optimization by adding some functional group using with the help of computers, so that we'll be able to get information. So that we'll be able to get compound that will be soluble and be able to do exactly what you want it to do. And some of the method of drug design is start from molecular dynamics, virtual screening, SAR, molecular docking, molecular modeling, KSA, molecular, all these things I've listed here. We are going to work on each of them during the course of this workshop. Now, when we're talking about drug design, there are 2 types of drug design that has been employed. Every form of drug design we have been doing are based on this. The first one is ligand-based drug design. The second one is called structure-based drug design. And if you look at the diagram I have here, there is a line that describes the structure-based, ligand-based. We are going to go into that deeply, but we have to understand what is ligand-based, what is structure-based. Now, ligand-based relies on the knowledge of other molecules that bind to a biological target. What that means is, if, for example, now, paracetamol, Tylenol, as you all know, that's the trademark name, is used to treat headache, right? And from biochemistry, how Tylenol works in curing headache is to bind to cause enzyme affecting the prostaglandin hormone to cure headache, right? So if you want to design a new drug that will do the same work like paracetamol, like Telenol, all you need to do is, based on the understanding of the structure of Telenol, you will take Telenol and you are going to make like a twin of Telenol. You will make another drug that will be similar in structure. For example, if this structure, I don't know if you can see this structure I have here. Can you see my cursor?
Yes, sir.
Yes. For example, if this, let's assume this is Telenol, right? And you want to make another drug that will have the same, that will also cure headache, right? All you need to do is design a drug, a compound that will have similar structure. Maybe this will be here, this will be somewhere here. It will not be 100%, it will be similar, but not 100% identical. Are you getting me? So by so doing it, if those functional group on Telenol are on it, there is probability that that drug is likely, that drug is possibly going to cure headache. And that is why it is called ligand based. I say anytime you see me using ligand, small molecule, they are all the same as drug. So ligand based drug design is designing a drug based on your understanding of an existing drug for a particular target. And that's why I use paracetamol as an example, Tylenol as an example. That if you are looking for another drug, you want to design another drug that will cure headache, then because you have understanding of the structure of Tylenol, you can use that structure of Tylenol to design another drug that will be similar in structure to Tylenol. Are you getting it? That will be similar in structure to Tylenol to design another drug that will have a similar thing. You understand? So when you are using ligand-based structure, you derive a pharmacophore. And look at this diagram I'm showing here. This is artificial intelligence at work. These are chemical similarities. There are this A and B. There are many chemicals. Are you getting me? This is machine learning, deep learning from artificial intelligence. So now, when you have this algorithm, are you getting me? You put this drug there to give you every example. If A is our Tylenol and B is another drug that has a therapeutic effect to cure headache, for example. By the time you put this in this algorithm, there are similarity search algorithm that will give you compounds that are similar in structure. You don't need to think. Instead of you designing yourself, you don't need to do that. Just go to some AI-based server. All you need to do, just put the structure of your Tylenol and the other drug, put it there. To generate hundreds of compounds that are similar in structure to your Tylenol. Can you imagine you feed in this, they pass through this AI algorithm and give you many hundreds of compounds similar in structure to your Tylenol. So once you have that, you've been able to identify some set of compounds that are similar to Tylenol, and you assume they are likely to cure headache. Even though your design does not stop there, but it helps you to know where to start from. Are you getting me? So when you have those similar structure, we refer to them as pharmacophore. Similar structure to a drug, pharmacophore. So once you do that, then you can now say, okay, let's do some ligand-based virtual screening, and you will be able to identify how many of them has similar characteristics with Tylenol. And you can say, okay, this is behaving almost like Tylenol, maybe in terms of physical property, in terms of structure, and you say, okay, this is likely going to what? Identify, this is likely to work the same way with Tylenol. But thanks to AI, you cannot design a drug similar to Tylenol on your own. You can do it, but it will take you days. But I'm telling you, in less than 10 minutes, if you make use of server like Zinc Farm, Swiss Similarity, they are AI servers. We are going to work with them. They will generate hundreds of compounds for you that are similar to a particular drug in less than 2 minutes. So that is ligand-based drug design. We design our drug based on the understanding of an existing drug for a particular disease, for a particular biological target. Now, structure-based now. Structure-based is not putting into consideration an existing drug. No. What structure-based is looking at the biological target we are focusing. For example, now, when COVID first came out, it will be difficult to use ligand-based for COVID to design ligand-based method to design a drug for COVID. Why? Because COVID just came out. There has never been any drug that has been used to treat COVID, isn't it? So in that case, what you need to do is to use structure-based. And structure-based relies on the knowledge of the three-dimensional structure of the biological targets. For example, if you want to design a drug against SARS-CoV-2 spike protein, you have to go and take the structure. For example, let's say this is the structure of the protein. This one in green and red is the structure of the protein. You look at the protein, you look at the structure of the protein, you look at the binding sites. How does the binding site look? How does it look? From the look and the understanding, the type of amino acid, the functional group, the charges on the amino acid on the molecule, that's your biological target, like this in green. We now give you information on how you can design a drug that will fit into that biological target. Are you getting it? And instead of you doing that manually, there are AI algorithms, computer softwares that will help you to study a protein. By the time you study the protein, you identify a point on the protein where your compound will bind to. That software will now go to list of compounds and will tell you, out of this compound, this is likely to bind to this protein. I believe you can see the difference between the ligand-based and the structure-based. I want you to have this understanding, because if you have this understanding, you'll be able to, by the time we are going, you will be able to link everything together. It shows you how everything moves. Ligand-based is based on the structure of an existing drug for a particular disease, right? But in structure-based is based on your knowledge of the structure of a protein, biological target, like spike from a particular disease. So once you have that, there are software like the Protein Data Bank where you can download the X-ray crystallography structure. And this structure can be obtained by X-ray crystallography, NMR, and the rest. And if you look at this diagram, let's assume this is a spike protein. This is an MMAA1 protein. Virtual screening, you'll be able to identify. Your software will tell you, okay, this lead compound is likely to stop this enzyme from functioning. Because the moment you stop this enzyme from functioning, you can cure the disease, like COVID. And once you do that, the next thing is to talk to a chemist, a synthetic chemist, to synthesize that compound for you. Once it is synthesized, then you take it to the lab and test your compound on it. It saves you time and energy. That is how it works. So now let's go. I just give you a paraphrase of a structure-based drug design. Let's go further. For example, if an experimental structure of a ligand is not known, this is another thing. What about situation whereby you don't have, maybe, like I said earlier, when you want to know a structure of a protein, people do research and they put it on Protein Data Bank. Is an NCBI, NIH web page, whereby people drop structure of proteins all over the world, not only in the US. But there are cases whereby there are some proteins that are not yet available on that structure. You understand? How do you do? What you do is you generate the structure through a method called homology modeling. Homology modeling is Once you know the amino acid sequence of the protein, you will generate the structure of the protein and you can use it for your drug discovery. Everything I'm telling you now, I want you to go back, watch this video. No, you take time to watch the video twice. You will understand exactly. But by the time we start the practical aspect of this whole training, everything we are talking about now will come to you in a clear picture. So I encourage every viewer of this to take time to look at this twice, at least 2 to 3 times, so that you have the understanding. So that by the time we get to the second other phase of the project, you will understand clearly. So when you can't get the structure of the protein on database, you can model the structure yourself using some AI-based algorithm like Swiss model, like Modeler, and many of them. Homology modeling, also known as comparative modeling of protein, refers to constructing an atomic resolution model of the target and an experimental 3-dimensional structure of a related model. So now, when you have a protein sequence, are you getting me? Maybe, for example, maybe let's assume we don't know how COVID-19 spike protein, for example, look like. We have no idea. But we have this amino acid sequence. Don't forget, every protein, I believe many of us are science students, proteins are made from amino acids: alanine, phenylalanine, tryptophan, glutamine, glutamate, lysine, the first 20 amino acids. You understand? Proteins are made from that, right? Now, when proteins are made from that, if you don't have the structure, all you need to do is to paste the sequence of a particular protein, maybe spike, for example, paste it on a server, and go AI server. Based on the information, is going to search from its database which other protein has a similar arrangement of amino acid to your own sequence that you want to build. Based on that information, you will be able to use, okay, there is a particular protein that has almost the same similarity amino acid sequence with your own protein. From that, it's going to use the structure of that protein to build your own, to generate your own structure for you. That is what is called homology modeling. That's why it's called comparative modeling of protein. If you use an existing structure and compare the sequence with your own sequence that you want to build and generate a structure for you, that's how it works. Now, so let's look at some of the advantages of computer-aided drug design. One of the advantages of CADD is time. I'm telling you, time. According to the FDA, it takes between 10 to 15 years for a drug to start from beginning and get to market. 10 years. But I'm telling you, with the help of computer-aided drug design, drug get to market in less than 5 years. Now, look at what happened during COVID. People were able to put together some drugs. They are able to repurpose drugs, build new drugs in a short period of time. And I'm telling you, many of them use computer-aided drug design. Even the vaccines, they do the same thing to generate all those things. It saves time, a lot of time. What about cost? It is very cost effective. Instead of you taking 15 compounds and start doing trial and error in the lab, computer-aided drug design CADD will let you know which of them is working, which of them is not working. Can you see that? Accuracy. There is high accuracy in this once you know what you are doing. And we are going to do that in our own project too. You will design your own drugs. Apart from the fact that we are going to publish, we are going to take them, we give them to lab to test for us that, okay, this is what Stephanie's group identified as possible cure of COVID. Can you please test it in the lab for us if it's going to work? And I'm telling you, I can assure you of 80 to 90% accuracy. That's how it works. So there is high accuracy when it comes to CADD once you do your work perfectly. It's also information about the disease. Using CADD, you can have much of information about the disease you are studying. Screening is reduced. You have reduced screening in it. You have reduced screening. It reduces the number of screening you do. Then you have database screening. There are databases that has over millions of compounds. Instead of you thinking, just make use of that over millions of compounds, you will see the one that is likely going to work for the disease of your interest. And there is less manpower that is required. I'm telling you, you can be in your house, you will be at your house, you alone, designing drugs on your laptop. Once you understand your programming language, once you understand your computer, you understand everything, you understand you have every information you need, it requires less people. And that's why by this, we are going to have three men group. There will be that is 3 to 4 people in each group, and you guys will work together and design something. So this is very, very interesting. Whether you are in biological science, whether you are in pharmacy, whether you are in engineering, anything, having a knowledge of this is good. Having a knowledge of AI generally in drug design and other aspects is good for you. And that is where the world is going to right now. I'm telling you that. And this, I even put together some of the drugs that are already on the market, not all of them, some of them that are already in market that have been clinically approved through CADD approach. All these drugs we are seeing, sequinavir is an HIV drug, captopril, all of these they are seeing, and many more. I just put few lists of them here for you. They were identified through computer-aided drug design since 1981. Can you see? As far as 1981 up to this moment, and some of these drugs are already in phase 2 clinical phase, and some of them are already past clinical phase, and some of them are already saving lives. Most of these drugs, all these ones, they are HIV. Most of them are HIV drugs, cancer drugs. They are already in market, saving people's life, and that is how they work. So that's what I want to. So that is just giving you an idea of what you are about to do so far. How we can apply AI through the computer-aided drug design to fast track the process of identifying new drugs, establishing new drugs, and make them get the market. You understand? And if you are interested, there will also be some further videos that you can do on this. And there is room for questioning. You can maybe send it through either direct message or through comments session. You understand? We address it, and the purpose is for everybody to have a clear understanding of what you are doing, so that at the end of it, of this workshop, you will say, yes, I've learned and I've gained something. Thank you. Do we have questions? Hello?